right, welcome back from break, everyone. We're just going to finish up this notebook and get started on the next part of the workshop before lunch. Okay, so the last challenge here was, recall that we created a scatter plot using X values from MyDF column 1 and Y values from MyDF column 4. Looks great. And the challenge was recreate this plot, but add 2 more parameters, type and call, so that we can create a plot with both points and lines colored purple. So let's go back to the help menu from the plot method and take a look at type. So if we want both points and lines, we have to set type equal to B, the string B. Now for call, we aren't given a whole lot of information, so this was kind of deductive reasoning a little bit. How do we turn a plot purple? So first we said type needs to be equal to B if we want to create a plot with both points and lines. Then if we want to color it purple, well, all you have to do is set call equal to a string purple. And this function is smart enough to interpret that string and map it back to an RBG code and turn the plot purple. So let's go ahead and run this. And putting that syntax into the chat as well. And now we have a nice plot with both points and lines colored purple. Does anyone not quite get what I did there? Any questions? All right, excellent. Well, in that case, that brings us to the end of the intro of our notebook. Of course, we can always come back to this to answer questions and discuss. But first, I'd like to show you how you can save your completed notebook. So please, first of all, go to floppy disk icon, save the notebook, and then go to File, Save and Export Notebook As, and HTML. And this will prompt you to save your completed notebook somewhere on your local computer as an HTML file that you can view later. And then the notebook itself, the actual .ipynb file will also be available on GitHub. So that's File, Save and Export Notebook As, and HTML. Great, and with that, I am going to hand it over to Amanda to start the debrief.
So we're going to go over RNA sequencing and data processing, again, hopping back and forth between the lecture and Jupyter Notebooks, where we're going to do the hands-on portion. Before we get into RNA sequencing, we're going to talk about some of the technologies that came before RNA sequencing to make RNA-seq possible. So the first one is one of my absolute favorite inventions. Back in the early 1980s, this cool guy right here, Harry Mullis, he had this idea for how to clone pieces of DNA, which we now know as polymerase chain reaction. And what PCR does, as some of you guys may already know, may teach, is we start with our DNA containing a template of interest. So we do need to know about the sequence that we want to look at, specifically the regions that flank the sequence depicted here in light blue, and depicted in dark blue is the region that we're interested in. A couple of other things we need to know about this is that the regions that flank our area of interest need to be conserved and need to be unique to the sequence that we're interested in. And that's to avoid off-target effects. So we want to identify regions flanking our DNA of interest that are unique, and then we design primers that are complementary to those flanking regions. And then we also supply this reaction with nucleotide triphosphates, so our A, T, C's, and G's, as well as our molecular machine polymerase to actually build new strands of DNA. And so what happens, this happens in a series of cycles. Every cycle has the same 3 steps. So the first step is a denature step, so a pretty high temperature here to split apart your double-stranded template. And then once those are split apart, it now exposes those conserved unique regions flanking that region that you want to clone. And so that allows the primers, which are complementary to those regions, to go ahead and bind or anneal to those regions. And then we have another temperature change again, and this temperature change will bring us into the extension phase of the cycle. And this temperature is ideal for polymerase to actually grab those nucleotide triphosphates that are in the reaction, starting in the area that's primed, and begin to begin to clone and build a new strand of DNA complementary to the region that we're interested in cloning. And so this, of course, happens on both, in this case, the antisense and the sense strand, or sense and antisense strand. Copies that are being made are antisense and sense. And then, so after this first cycle, what we started with is one copy of our double-stranded DNA template. What we end with is 2 copies of our double-stranded DNA template. We then repeat these cycles multiple times. And then each time the same thing happens: denature, annealing, extension. And so we go originally starting from 1 to 2 in our first cycle, and then that same thing happens to each of these 2 double-stranded templates in our second cycle, which will give us 4 copies. And then each of those 4 copies will then make copies in the third cycle, giving us 8. And so depending on how many cycles we run, we'll end up with 2 to the n copies of the region of interest that we want to clone. So this was super cool, so cool that Kary Mullis won the Nobel Prize for this invention, and it has been built on ever since. So one of the limitations of this in the context of trying to understand what's happening in gene expression is that this isn't really quantitatable, right? So we don't really know how much material we started with or how much material we started with in one sample relative to another doing this. So this technology was expanded upon. I believe quantitative PCR came about somewhere in the 90s, early to mid 90s. And what happened here, in this example now, we're starting with an mRNA. So this is going to be useful to us if we want to look at gene expression being depicted starting at the message RNA level. And so we'll take our message RNA, we can then convert it into complementary or cDNA using a reverse transcriptase. And so now we have our cDNA, which again was derived from our message RNA. And we know, of course, with and from this mRNA, there's going to be a certain gene that we're interested in. And so we will know the sequence of the gene that we want to look to see the expression of. And this time we'll go through and do PCR again, as I described in the previous slide, but this time we're going to add another reagent called CyberGreen. And what CyberGreen does is it'll bind to double-stranded DNA, and it will, with being excited by a light source, it'll produce a fluorophore that we can then take a picture of. And so what happens here is every time a new double-stranded template is made, it gives off a light signal. So, and then once we go through the denaturation step, that double-stranded DNA is then separated. And so the signal is no longer there. And then when you go through the next cycle, you have double-stranded DNA again, make another signal. And of course, as you go on, because you're making copies of the region that you're interested in, you are seeing an increase in the light that's being detected from the CyberGreen fluorophore. And what's nice about this is, of course, at the beginning, you're going to start with some background signal because you start with your cDNA template in here. But now we can start to use this to compare the expression of a known gene between 2 different samples. So if we have, in our case, a sample that was flown in space and a sample that was kept on the ground, and we're really curious to know what the expression of gene A was in those samples, if we isolate the same, if we isolate RNA and make cDNA, and then run this assay on that cDNA identifying gene A, if there was more gene A in our flight samples at the start, that signal is going to be brighter faster than our ground control if our ground control had less of gene A in it. And so now we can start to do a semi-quantitative comparative analysis of which sample started with more of the gene that we're interested in. This is great, but as I mentioned, cyber green binds to double-stranded DNA, so it's not specific. So any double-stranded DNA that exists, it will bind to, and so that has the possibility of having off-target effects. So another assay was developed for quantitative PCR called Tapman. And so what Tapman does is, in addition to having a conserved region flanking your gene of interest, the gene that you want to know the expression of, so your forward primer and your reverse primer, you also have to have a conserved region somewhere in the middle of your gene of interest that is, again, unique to that gene. And then we design a probe called the Tacman Probe. And this probe, which is complementary to the middle of your region of interest, contains a fluorophore on one side and a quencher on the other side. And so what happens is when this fluorophore is in close contact with this quencher, like it is at the start, when it kneels down, it inhibits that fluorophore from releasing a signal. And so now when you run your PCR, you give it a special kind of polymerase called Taq polymerase, which has 5' endonuclease activity, so it can cleave base pairs at the 5' end. So now when this Taq polymerase goes during that extension phase of the cycle, and it starts building your nascent strand, building your new strand from your template, once it reaches that Tacman probe, its endonuclease activity is going to be activated. It's going to cleave that fluorophore, which is going to allow it to be far enough away from its quencher to give you a signal. So now, every time a copy of your region of interest is made, it's going to release a signal that you're going to capture and measure the amount of light. And so again, similar to what I described in cyber green, the more starting material you have, the more quickly your signal is going to get brighter. And so just to kind of show you what those data look like for those of you who are unfamiliar with it, down here on the x-axis, we have the number of cycles. And then over here on the y-axis, we have increasing in fluorescence. And so as I mentioned, you'll get some background signal initially. And then what you can see here is, I'm assuming here, each one of these lines represent a sample. And I assume that you see that there's multiple colored lines kind of overlapping each other. This is often run in triplicates just to be really confident that you're seeing the signal that you're seeing. So I think that each one of these are triplicates of a different sample. And so as it's building and going through the cycles, what you'll see in the signal is this linear amplification phase. And then at some point, there will be so many copies of your region of interest that it'll be so bright that it'll saturate the detector. And so you'll get this plateau here. So now if you want to compare what was more expressed at the start, well, samples that had more starting material are going to get to this linear amplification phase with a lower number of cycles than something that had less of the starting material that you're interested in. And so now you can do is you can pick a region or a level of fluorescence within this linear amplification phase. And you can say, okay, this sample here, it took 17 cycles to get to this fluorescence, whereas this sample here took 21 cycles. And now you can start to use that to see which sample had more starting material or which sample had more of your gene of interest expressed or less. And so this allows us to do quantitative PCR. The other thing I should mention is I've been discussing right now kind of comparative analysis of the level of expression in each sample. You can also have a sample with a known amount of your starting material gene. And if you have a known amount of your starting material, you can add a known concentration and run this assay, and that will build a standard curve. So then you could know, oh, when I put so many molecules of cDNA into here, it came off at cycle 14 is when I was at my linear amplification phase, or it came off at 19 when I had less amount of my control material. And so you could build a standard curve to kind of give you an idea of the number of cycles relative to the concentration of your starting material. So that's one way where you can actually have like a real or close to real quantitation of how much starting material is there, as opposed to just a comparative analysis like I described. This technology comes with limitations. Actually, before I continue, I just want to point out, let me go to the previous slide here. I just want to point out that everything that I show, I have links to. So if you guys want to learn more about that stuff, I also in the PCR and qPCR slides, I have links to videos if you guys want to see videos of how that works, if things aren't clear. Or I can try to explain something differently if it's not clear. So we do have some limitations still with quantitative PCR. So remember, you have to know the sequence of your gene that you're looking for in order to evaluate it, because you have to design those primers to be able to look specifically at that gene. So only genes that are known and annotated can be evaluated. There's the potential for non-specific binding, specifically, especially with Cyber Green, because it has a double strand, it just binds to anything that's double-stranded. It requires primer optimization. You need to make sure that your primers are annealing properly and are amplifying the region that you expect. That could take some time. If you're doing the Tacman method, you need to find 3 conserved and unique regions for the forward primer, the reverse primer, and for your Tacman probe. I didn't go over this, but I should have. So in order to control for the amount of starting material that you added for each sample, you also always look at a housekeeping gene or a gene that you know is not going to change in expression between your samples. So that way you could use that to normalize your data with to control for your starting material. And so you do need to know those housekeeping genes as well. Absolute quantification of the target gene requires a standard curve, as I just talked about, which could increase the likelihood for issues if the standard curve is not properly generated. It's limited in the number of samples and number of genes that can be evaluated simultaneously. So I've done this in as many as a 384 well plate without a multi-channel pipetter, which was an absolute nightmare. So it is limited in the number of samples and genes that you can evaluate at the same time, and also you have to run them in triplicates to make sure that you are confident in the signal that you're receiving from your samples, which further limits the N number that you can evaluate at the same time. And then I just encourage you guys to think about other limitations of this technology. This brings us to 11 o'clock, which is our lunchtime. And so I'm going to pause here, and when we pick up after lunch, we'll start with any questions that you guys might have about the material that I just covered. But I do want to be cognizant of your break. So please go ahead, take a break, take your lunch, and I will see you guys back here at noon Pacific time.